Hello, children. This is Dr. Rupali from Dr. Amina classes और हम अब चैप्टर 12 करेंगे फास्ट रिवीजन बायोटेक्नोलॉजी एंड इट्स एप्लीकेशन. 3 critical research areas of biotechnology क्या हैं? Providing best catalyst, creating optimal conditions and downstreaming. तीनों देखिएगा, कभी कभी डायरेक्ट 3 मार्क्स का क्वेश्चन आ जाता है. Then applications in agriculture of genetically modified organism. Genetically modified plants useful in many ways. कैसे? BT cotton, Bacillus thuringiensis बैक्टीरिया से BT टॉक्सिन लेते हैं. DNA, उसके DNA से क्या बनाते हैं? टॉक्सिन, BT टॉक्सिन, जिसकी उसकी जीन्स को बोलते हैं क्राई जीन्स. वो टॉक्सिन जिससे बनता है. सो cry1AC, cry2AB, cry1AB, both these are used to kill larvae of cotton bollworms, and cry1AB for corn borer. This can be asked. Now there is a technique where this toxin becomes active in alkaline pH in the gut of larva, and it causes holes, so the larva dies. So it is used as to kill the insect larva. Now for pest resistant plants, very important technique, RNA interference, and this technique has been used. Double stranded RNA is used to prevent the transcription so that no proteins can be formed, and this has been successful in killing the nematodes Melidogyne incognitia, which infects the roots of tobacco plants mainly, and huge losses are there. So this is prevented from this technology. Now applications in medicine, very important application, genetically engineered insulin. Figure 12.3 is given. You have to go through the 3 amino acid chains, and 1 C peptide is not present in mature insulin. So the insulin was prepared in 1983 by Eli Lilly Company of USA, and it is being sold in market as Humulin. Now one more important, always ask, gene therapy. Now gene therapy in 1990 was done in a 4-year-old girl. She was having ADA deficiency, that is an enzyme, adenosine deaminase, which is very important for our immune system to function properly. Now you have to learn this gene therapy, what was the how it is done. But one more question is asked, that what is its permanent cure? So that you have to do from NCERT book. This is important. Then diagnostic tests like ELISA, enzyme link immunosorbent assay. The principle is antigen antibody interaction. This is a diagnostic technique, so you have to do this. Then animals में बायोटेक्नोलॉजी का एप्लीकेशन, ट्रांसजेनिक एनिमल्स, उसकी डेफिनेशन. और बहुत सारे एनिमल्स पे ट्रांसजेनिक एनिमल्स बनाए जाते हैं, लेकिन 95% mainly are mice. How can man benefit from such modified transgenic animals? उसका है पहले नॉर्मल फिजियोलॉजी एंड डेवलपमेंट. सो क्या है इसके अंदर? नॉर्मल फिजियोलॉजी एंड डेवलपमेंट में क्या देखते हैं कि ट्रांसजेनिक एनिमल्स कैन बी स्पेसिफिकली डिजाइन टू अलाउ द स्टडी ऑफ हाउ जीन्स are regulated and how they affect the normal function of the body and its development. Next, study of diseases के लिए भी हम transgenic animals use करते हैं. Transgenic model of animals exist for study of diseases like cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. This is to understand how genes contribute to the development of diseases. Next is for biological products. Now this is important. Few important examples इसमें हैं. कैसे? Human protein alpha 1 antitrypsin, it is obtained by this technology to treat transgenic animals, to treat emphysema. So this is being asked many times. In 1997, first transgenic cow Rosie was um, obtained. Then human protein enriched milk. How much protein? 2.4 grams per liter. And that was human alpha lactalbumin. So that was nutritionally a more balanced product for human babies than natural cow milk. So these are all examples are important. Next, vaccine safety. Transgenic mice are being developed. पूछते हैं कौन सा एनिमल? Mice are being developed for use in testing the safety of vaccines before they are used on humans. ठीक है. Lastly, chemical safety testing, toxicity of drugs. It is used for testing toxicity of drugs. The animals are exposed to toxic substances and their effects are studied. So this is how transgenic animals are being used for the benefit of man. Now, because of this, we are playing with the DNA, genes. So there arose many ethical issues. So to control that, Indian government has set up organization GEAC, पूछते हैं full form, Genetic Engineering Approval Committee, which makes decision regarding the validity of GM research and safety of introduction of GM organisms for public services. So they ask what is the function of GEAC. Now comes the two terms are there, biopatent and biopiracy. So biopatents, there are some crops, some plants which are indigenous, इंडिया के ओरिजिनल हैं, वो इंडिया में ही कई सालों से उनको यूज़ करते हैं, उनकी मेडिसिनल वैल्यू, लेकिन उनको चुरा करके उनका पेटेंट ले लेके दूसरा कंट्री कोई उसका पेटेंट ले लेता है कि यहाँ हम बेचेंगे, हम उसको, और जिस कंट्री का वो असली में प्लांट है, वो जहाँ वो ओरिजिनेट हुआ है, उनको उनके संग ये धोखा है. सो वो जो एक कागजात होते हैं इसको लेने के लिए कि हमारा प्लांट है, उसको बोलते हैं पेटेंट, और क्योंकि वो प्लांट का या लिविंग ऑर्गेनिज्म का है तो बायो पेटेंट. सो ऐसा गवर्नमेंट हर जगह की ये देती है. और हमारे यहाँ बासमती चावल हमारे यहाँ का ओरिजिनल प्लांट है, और उसको यूएस गवर्नमेंट ने उसका पेटेंट 1997 में उसके राइट्स ले लिए थे. So that was wrong and this is a type of biopiracy. So biopiracy रोकने के लिए भी Indian Parliament has recently cleared the second amendment of the Indian patent bills that take such issues on consideration, including patent terms and emergency provisions and research and development initiative. So biopiracy में ये रोकने के लिए.